Fluorescence Resonance Energy Transfer by Regina Tanchez. Fluorescence resonance energy transfer is also referred to as FRET. FRET is a physical process that is a distance dependent in which energy is transferred non-radiactively from an excited molecular donor fluorophore to another acceptor fluorophore by means of an intermolecular long-range dipole-dipole coupling. FRET can be used as an accurate measurement of how proximal molecules can be at an angstrom distance. It is efficient if the donor and the acceptor fluorophore are positioned within a Forster radius. A Forster radius is the distance at which half the excitation energy of the donor fluorophore is transferred to the acceptor fluorophore, typically between 3 to 6 nanometers. The FRET technique is used because it's a study of molecular interactions inside living cells with an improved spatial and temporal resolution. It is sensitive and a broader range of biological applications. FRET occurs when the donor channel signal quenches the acceptor channel signal and it will be synthesized or increased. FRET studies of donor and acceptor fluorophores come from FPs. FPs are fluorescent proteins and are a class of autofluorescent proteins. GFPs are green fluorescent proteins. All of the fluorescent proteins are carefully considered in the selection as workable FRET pairs. GFPs-based FRET imaging methods have been a key in determining the functional organization and compartmentalization of living cells and the tracing of movement of proteins inside. FRET microscopy relies on the ability to see the interactions of labeled molecules in single living or fixed cell through the capture of fluorescent signal. In this experiment, the discussion was that there were various methods that had proven to be reliable for measuring protein interactions by FRET microscopy, highlighting the advantages and limitations of each method, and the most useful FP candidate for FRET microscopy. In this experiment, they discussed the selection of FPs for a different FRET method, identifying the most useful FP candidate for FRET microscopy. In the recent success in the expansion of the FP color palette, it offered the opportunity to explore new FRET pairings between the FRET and FP. In the figure below, we see that the spectral imaging of a cell producing the venous fusion protein. Image A, the cell was illuminated by the donor excitation wavelength and spectral measurements were acquired from the ROI indicated by the red box. The linked venous fluorophore was then photobleached using the 514 nanometer laser line in the ROI indicated by the yellow box. This resulted in more than 70% decrease in the venous signal. In image B, the spectral measurements were then required under identical conditions to the first from the same ROI red box, and changes in the donor signal were measured. The dashed line indicates the gray level intensity of the donor signal before A and after B acceptor photobleaching. The results showed that the dequenching of the donor molecules involved in FRET, leading to increased fluorescence emission from the donors. Finally, since photobleaching of the acceptor fluorophore is irreversible, the photobleaching FRET or PB FRET approach is an endpoint assay that is not amenable or dynamic for dynamic measurements. Importantly, PB fret does provide a method to verify fret measurements made using other techniques. Some of the drawbacks of this experiment are that the primary weaknesses of spectral bleed-through or SBT correction approach is that it relies. The question I have is, how can one distinguish between a radioactive or non-radioactive fluorescence resonance energy transfer? These are the references of the papers that I used for my presentation.